In previous lectures, we have seen that we can model the behavior of simple genetic circuits by using an approximation that is grounded in Boolean logic. So in other words, we simplify a whole biology that is behind gene expression by assuming that genes either are expressed or not. This is an approximation. We'll keep asking ourselves to what extent this is a good approximation. But I just wanted to remind that actually we are compressing a lot of details in the picture we're using when we write down genes as kind of a simple scheme like this, right? Something that can be expressed, that has some control zones, promoters, etc. And that leads to an RNA, which can lead to a protein, right, with some given rates. And of course, rates themselves capture a lot of information. It's compressed there. This basic scheme actually is what we say as a model. And the model simplifies the whole picture, but I wanted to stress something. On the one hand, that there's a number of details about the molecular mechanisms responsible for expression and translation. So how things eventually go into proteins that allow cells to perform functions. But I also want to stress something. This is also a model, okay? When I write down this on the blackboard, what essentially I'm saying also is that I'm compressing reality by ignoring more details, like DNA is a three-dimensional molecule. It has a compact form in space, a non-trivial one. There's a spatial localization, etc., etc. We are always working with models. So we move into this picture at some point where you can actually write down differential equations and treat that as a continuum problem. And in the more recent lectures, what we actually did was to imagine that genes are on-off devices and that they can connect to each other. And for example, we consider the idea that circuits that are very well known in engineering, one extremely simple one, almost trivial, is a circuit like this, where you have, for example, a light bulb. A light bulb is going to be my output, whether or not it's lighting. I have a source that allows electricity to move, and then I have two switches. And for this particular model, it's very clear that there's only one solution that allows electricity to move around, right, and make the output be a 1. 1 for me is working, okay? So for this particular system, we saw that actually this defines what is called a logic gate, an OR logic gate. In other words, if this is my switch 1, my switch 2, and I assume that 1 is closed, 0 is open, this circuit essentially gets compressed into a gate, okay, that is represented in a compact form in this way, and in a logic way as a table, okay, where all my combinations, okay, are explicitly stressed here. And for this particular gate, unless the two switches are open, the output is 1. And that would be the truth table of the logic gate. That's a very simple way of putting this. And we show that we can actually design a simple genetic circuit, right, where gene-gene interactions are introduced in a very simple way that allows actually to build this particular logic gate, okay? This can be generalized. We saw several important things, like for example, that there are special gates like the NOR gate and the NAND gate that allow to build any circuit you can think, right? And in electronics, this is a standard. And we want to make a step forward now. The circuits we consider are circuits that usually are saying feed forward, right? This will be a very simple circuit The logic gate essentially says that there's an information from the input to the output in one direction, okay? And the first thing that we want to do is how I can generalize this, right? We are not going to spend much time because, as you will see, it's really straightforward. How do you build more complex circuits? And the way of doing that is the following. This is a function, a discrete dynamical system of a class where my logic gate can be thought as a function from the set of pairs, of binary pairs, to a 0 or 1, right? Remember that we use this notation for our set of possible states. Okay, so what if I want something more complex? For example, imagine that I want to do something like I have an AND gate, okay? I have another AND gate here, all right? So now I'm playing with 1, 2, 3 inputs, and in this case, just as an example, right, the same gate. Okay, how do I deal with that? This is again a feed-forward system, right? I have inputs and I have one output. I could have more outputs, right? I'm not going to go into that. But as you can see, now we are moving in now another group of functions that map this set into sigma, right? And this, of course, can be scale up. And that actually is one of the big successes of technology, in particular in information technology, when it was possible to actually move from single units into really complex systems, right? Thanks to two things. One, combinatorial logic. We can actually combine an arbitrary number of gates, right, of any class, to virtually implement anything we can think about, like a computer, right? And the other thing was miniaturization, potential of making things smaller and smaller and smaller. So in just some decades, things scale up from a few transistors or few gates into millions. And that's a question that we're going to ask ourselves in the context of synthetic biology, right? Can we do the same? Right, how can I manage to actually see what's the outcome of that? And it's extremely simple. You actually can follow the same logic. You know that from the units, right, my AND gate, if I have, let's say, in general, A, B, and out, remember that the AND gate only allows an output to happen if the two inputs are ones, right? So for the rest, what I have is this. That's the only case that is, say, favorable, right? All right, so I have a larger table. So now what I have is my input set is made of 3 numbers, right? Boolean numbers. And my output set is limited to a single Boolean variable. We can write down this table. We need to organize, as usual, orderly, right? If it was in integer numbers, will be 0, 1, 2, 3 until the end. As you can see, it's 8 cases, right? In Boolean, in binary numbering, what we have is, that's my trick to build it, right? I'm repeating this here. This will be 0, 1, 2, 3. I'm repeating the same here. This is just a simple way of remembering how to do it. And then I put zeros and ones. Remember, if you don't have that in mind, that the mapping from binary to our standard decimal numbering approach is that you take any value here. Let's say that this is, the numbers here are A0, A1, A2, right? The corresponding number here in our standard numbering will be A0 2 to the 0 plus A1 2 to the 1 plus A2 2 to the 2, right? For example, here you will have 1, 1, 1. That would be 4 plus 2 plus 1, 7, right? My last number will be 0 here, and you can show that this is just the mapping, right? So for me, in binary basis, that would be this number here will be 1, 1, 1, or in general will be A0, A1, A2. That's the way we translate things. Okay, so how can I now build this? Once you are more or less familiar, it's straightforward. It's very simple. One thing you can do, if you want, right, as an intermediate step to see how it works, is the following. Let's call this out 1, right, the output of this, and let's call out the final output, right? In this way, it will be very simple to see how it works. You can see everything that happens in each gate depends on the inputs, right? What's going to be the output 1 from A1, A2, right? So I just need to look at this subset now, right? And I can see, I don't not have to go case by case, because actually there's only one favorable case. 1, 1, 1, 1. These are the only cases where the output here, right, will be 1. All the rest are zeros, right? So 0, 0, 0, 0, 0, 0. And now the output I'm looking for, this one, is a combination of this number and this number, I3. So I just need to look at these numbers, right? And again, there's only one favorable case, which is one we have 1, 1, right? All the rest are just zeros, okay? Which kind of makes sense, because there's a very limiting gate. You can generalize that to anything you want. We can actually see the result of that in more formal terms when we think that the output is obtained by a logic chain of things. It's a combination of I1 and I2, and remember that this is written like this, okay? This is for my AND. Can also be use this expression, and I3, right? And that allows also to easily make the calculations, all right? So for you, for example, let's consider the case where I have OR gates, right? It's a very simple case. Or you can also consider other situations that combine more complex circuits, right? For example, we could actually think in terms of our interesting NAND gates, right, that combine into this, right? For you, a simple exercise, and you can see that it's very easy to generalize, right? Remember, NAND gates are very interesting for us because just with NAND gates we can build anything, okay? All right, scaling up this idea is very simple, right? And the next step we are going to do is follow a more interesting path, which is what happens with genes interact in non-feed-forward ways. What happens, you can actually implement this particular system using, that's also an exercise for you, a genetic system, right? What kind of coupling between genes should I use, right, in terms of regulation to actually implement this kind of circuit, right? You'll see, very easy. We'll go into something more interesting. What happens when you have a feedback? So when you actually have something as a product of the expression pattern that maybe, for example, regulates my own activity. What happened then? And that's the really interesting part of the story, because that is what actually allows cellular biology to be extremely rich in many ways. And a very important question for you, right, in terms of bioengineering. You might think, okay, combinatorial logic seems to be something that is straightforward. So I can just need to think how to connect things to each other, like in a circuit. My question for you is that the case? Because if you think about, there's a really fundamental difference between the implementation that you perform in electronics when you connect gates and what you require to do when you engineer circuits within cells. Think about what is the main difference for that. So what happens when we want to actually get a bit closer to reality and see how genes actually talk to each other, right? It would be easy if biology was just genes expressing themselves and looking at who is expressed, and from that, understanding what's the behavior of the system. That was the old idea, the reductionist idea in biology for many years. At the beginning of molecular biology and for decades, the picture was that I do know the genes that are expressed, so I can actually infer the behavior of the cell and the behavior of the organism, right? That was a complete failure, but it took a while to realize that. So again, the thing is, I have an underlying scheme, right? In more advanced courses, you will see that you can actually treat these details by using more sophisticated mathematical models. We keep, for this freshman course, into the simplest way of drawing the models, right? So let's consider one particular example. We already seen that you have two genes, for example, that express two different proteins, and they form a heterodimer. They can actually, for example, repress a third gene, right? And there's a feed-forward circuit. But now consider the following idea. Imagine that my genes implement some kind of, for example, a NOR gate, right? Two genes interact on another gene, and to make this simple, imagine that I have one gene that is implementing a NOR gate that affects another gene that also implements a NOR gate, and they are connected to each other, right? Remember that this in our diagrammatic description is expressed in this way, right? And that would be gene 1, let's say gene 2, yes, and gene 3, right? So for me, gene 1 receives inputs from the other two genes, 2 and 3, right? And its response, right, whether or not it's active or inactive, okay, will be the output of this, right? And we can figure out how to do that in molecular terms. Now, what I want to see is how can I actually understand the dynamics of the system when you have these interactions, right? These two genes are inputs for me, so my arrows just indicate the inputs from one to the other, and I have a closed circuit like this, right? So I don't have any more feed-forward system, inputs, outputs. The inputs and the outputs go together, okay? So this dynamical system, right, can be described in what way? Well, first of all, you realize that now what I have is Boolean dynamics, right? Not Boolean function, but Boolean dynamics, where I have inputs, have this three-dimensional system from a set of states. Each gene has two different states. I have three genes to the same set, right? Because what I'm doing now is acting on in time, right, from a set of states. Let's say S1 time t, S2 time t, S3 time t. For simplicity, I'm assuming that time is going to be discrete, right? 1, 2, 3, right? So that somehow I will know the state on time t plus 1 of my variables, right? So the states of gene 1, gene 2, gene 3 are going to be expressed as binary variables, right? So any of these numbers is going to be 0 or 1. But now I have a dynamical state. So how can I work with that? How can I build the tables? And it's very easy again. Now I am having, let's say, S1 time t, S2 time t, S3 time t. Now I have a larger table, okay? Because I need to map each 3 states into my new 3 states, okay? We'll do this example, and you'll see that this is, again, really straightforward, right? There's no much magic here. The magic comes from something else. So again, I write down my table. Okay, put this ordered in this way, and this to complete what is there. Okay? Okay, now, the state of any element in the system is fully determined by the inputs that it receives from the 2 elements that send inputs to it, right? And we know that the NOR gate is something simple, right? NOR gate, let's remember. The NOR gate means that if I have these 2 inputs, that's my output. Remember that the OR gate will be 0, 1, 1, 1. If one input is one or the other, right? It will be 0, 1, 1, 1, 1. NOR means I say the contrary, right? So that would be the NOR gate, right? There's only one case that gives 1, right? It's when the 2 inputs are 0, okay? Now, having my table, right? How can I actually translate that into this particular system? Well, it's easy to do, and actually you can do it the following way. Imagine I have my first gene, right? What's going to be the state at the second iteration of my modeling? So I only need to do the following. That's the set of states of my input genes, right? So I just need to look at their states, right? And then decide what's my next state, right? In the next iteration, right? Essentially by using the NOR gate logic. So I take here my inputs from these pairs of states, and that decides my output here, okay? So what's the only favorable case when I have a 1 as an output, right? Every time that my inputs are zeros, which is this case here, right? That's one favorable case, another favorable case. In these 2 situations, I have a 1 here and a 1 here, consistently here. The rest are zeros, so I just need to put zeros in my table. The same happens with the other numbers, right? Now I'm looking at the second gene, okay? What's the state in the new iteration? So I need to go now into the neighbors, right? So the first column and the third column, and look again at my table, right? My NOR table. What are the favorable cases when I want to have a 1 as an output state? And you see, 0, 0, that would be 1, right? 0, 0, that would be 1, okay? The rest, right? I'm not going to find any other place a 0 and a 0 anywhere because it's only 1s here, for example. So here I'll have these states, right? And there's a final situation for my third gene, okay, when I just need to look at which pairs here are 0, zeros. And I can see 0, 0, 0, 0 is the only situation. All the rest are combinations where I have a 1 somewhere. So only here and here I'm going to have this, okay? All right, so that's the table that results from that. And how can I express the potential dynamics of my system, right? I can do it in a couple of ways. The first one is to use my table to see what kind of final states do I find in the system. And it's easy to do. You just need to just follow state by state. States means now the 3 numbers, right? That's one state, like 0, 0, 0. And this, as you can see, 0, 0, 0 goes into 1, 1, 1. Okay? All right? Now, I'll take this, 1, 1, 1, right? That's my new state. And see what's going to happen in the next step. I see that 1, 1, 1 goes into 0, 0, 0, right? So, see what happens. I have an oscillatory behavior, something that we haven't seen before, right? The fact that you have feedbacks provide the source for something really important, which is cyclic dynamics, right? Attractors that are not just single states. If you keep doing that, right, you see that 0, 0, 1 goes into 0, 0, 1, so into the same state. So that would be another attractor here. 0, 1, 0 goes to 0, 1, 0. So if you are there, go there again. 0, 1, 1 goes to 0, 0, 0. And let me write down this here. 0, 1, 1 goes here. Okay? I'm marking because I have to complete everything. 1, 0, 0 also goes into 1, 0, 0. Right? So these are very simple attractor states. 0, 1, 0, 1, 1, 0 go into 0, 0, 0. So I also have these 2 states that go here and these 2 states that go here. Okay? So I have 3 attractors in my system. Oops. Excuse me. These attractors here and these attractors here, right? Once we get there, we remain there, right? Important. Now, discrete dynamics allows a dynamical state where you have oscillations. So, another way of representing that, right, my attractors, is to use the Boolean Hypercube, right? Remember that we have mentioned that, and also that we use that in fitness landscapes, right? It's a combinatorial possibilities of a dynamical system. This is not evolutionary dynamics, this is dynamical systems. I put here my, 0, 0, 0, 1, 1, 1, and let's locate the other values in order. And now I can use the Boolean cube to actually draw all the trajectories, right? On the one hand, you have the simple attractors, right? 0, 0, 1, it is here. I'm going to indicate this with a little circle to say this is an attractor state, right? I have the 0, 1, 0. Okay. I have the 1, 0, 0. Right, my simple attractors. And then I have a cyclic attractor, so something that involves 2 states as part of the final state of the system. One is this one, and the other is this one. Okay? I go from here to here, and I go from here to here, right? In an oscillatory way. What happened with the other? The other form what we say the basin of attraction, right? Those states that are not in the final attractor, but converge into that. And we see that 101, 011, 110, the vertices that we haven't not yet covered, go into one of the attractor states, right? This goes here, this goes here, and this goes here, right? All these states that converge into that are what we call the basin of attraction, that go into that particular state, okay? This, as you can see, is very systematic. You just need to know what functions connect, in this case, pairwise interactions between two genes and some target, right? And build the table. Of course, we limit ourselves into three-dimensional systems. You can see it also becomes really complicated and clumsy to go into more states, right? This rapidly grows in combinatorics, and it's something that we will discuss at some point, right? How biology manages combinatorics and how we, if we want to do bioengineering, can manage combinatorics, okay? So let me suggest for you an exercise that is a different kind of dynamical system that is extremely interesting. It's been really important in synthetic biology. It's a circuit, it's called a repressilator. And then you can actually map into this formalism, right? I'll tell you what's the basis of the circuit, right? The basis is the following. Imagine that I have a gene, right, that leads to a protein, okay, that's my gene 1. This protein is a repressor of a second gene, right, which also produces another protein, let me draw it like this, which is a repressor of another gene. And this gene produces another third protein, right, that is the repressor of the first, right? No wonder why the name of the circuit is repressilator. So this is a closed circuit, right? Remember, it's not just one signal into another signal, it's a loop, right? So this is a very interesting problem because it provides the source of something that is, again, very important, both in technology and in biology, right? In technology, we used to use something fundamental in electronics to actually make possible that this implementation from one set of discrete states to another happens rigorously synchronized in time, which is an internal clock. We have a clock that actually allows that to have t plus 1. So in biology, we have rhythms, right? Internal rhythms that actually can define a lot of the dynamics, and in particular, how it synchronizes with external things, right? And also are the basis of synchronization of many cells together to have responses to all kinds of external stimuli. The repressilator is an instance of that. How to synthetically define a circuit that, when you analyze the dynamics in time, we don't have the tools in this course for doing that, but you can actually map that into differential equations, right? And see that, very simple put, what essentially you have is that the circuit displays, if you look at the activity of any of the genes in time, displays an oscillatory behavior, right? So they synchronize in time. So we don't have the tools for using the differential equations, but you do have the tools now to actually build the circuit, right, in terms of Boolean logic, and to show by using this Boolean cube that indeed there's, in particular, is a large attractor that connects several states into a limit cycle, it's called in dynamical systems. And that, again, is going to be serves us to build more complex problems, right? For you, last thing I want you to think about is the following problem. Imagine that you have a system, you have to think about how you will implement that, where the state of a single gene, for example, gene 3, right, depends on the majority. Majority meaning what? If the majority of genes, including himself, is zeros, right? So I put myself in state 0. If the majority is in state 1s, I put myself in state 1, right? What would be the truth table for this? A hint, you don't have even to think much into Boolean gates. It's much easier. And more important, what is the outcome of that? What kind of final attractors do I have in my system? As you will see, this problem is connected with something that we already saw before, which is the problem of symmetry breaking.